Dear viewers, Assalamu alaikum. Today we will discuss homology modeling of protein and also the estimation of quality of protein structure when it is in 3D format. So in this homology modeling we will use Swiss model tool, which is the tool from Swiss Institute of Bioinformatics, and this is fully automated protein structure homology modeling server, and it is online server which is available for the homology modeling. So we will go to start. So here we will insert our FASTA sequence of protein, which is our target protein, and we will analyze the other proteins which are similar to this protein. We can also upload target sequence file. So for protein, first we will use this one. So this is our protein, which is crystal structure of human carbonic anhydrase 1, which is in complex with benzene sulfonamide, and it is my thesis research in which we have crystallized benzyl aminoethyl uridotyl benzene sulfonamide inside the active site of human carbonic anhydrase 1. So this is our publication in 2020, and we have deposited this crystal structure and some others also in PDB. So from here we will download this FASTA sequence. Now we have FASTA sequence of this file, which is here. Double click on this, will be selected, copy, and then we are going to paste in this Swiss model. So here we are going to paste, or we can also select file from the source, and then we can also take this file. Like here I am going to show you that we can also upload target sequence file. So we can also upload this file if we have this file, which is this one, and I am going to save this file on the desktop. So we can open this file. Now this is again the same file, which is the sequence of this one we have pasted here. Now we will go to search for templates. We can write here the title, project title. Like here I am going to write here is 1 or 2, email address, then we will get email of this, and then search for template. Now our results are running here. So this is our project which is running here, and the sequence alignment is going to occur. We will get here results for our protein after some time. It will take some time, and after analysis it will display the results on our screen. So we are waiting for this. Now the results are here. So our compound 24 templates matches our protein, this protein, and here the model is present here. So this is our protein with different amino acid residues. So we can make a cursor to check that which amino acid is present at which position. Like here it is histidine 94. So these are our results, and this protein shows that it is the identity is 100% and the method is X-ray crystallography and 1.5 angstrom resolution. It is also in monomer state, and the ligand is zinc and 3TV. So these are the ligands which are present inside this protein. Like here it is this tetrafluoropyrosulfonyl benzene sulfonamide. So in this way, the other structure which is also similar with 100%, but this ratio is 0.98, and the resolution is 2 angstrom, also monomer, and 2 zinc residue, and along with this ligand is present inside this protein. And this is the ID of this PDB ID of this protein. So here we can also check this protein, or also we can check this protein. So these are the matching proteins, or we can unselect these proteins. So all these are 24 proteins which have similar sequence with this protein. And we can also check the quaternary structure sequence similarity. So here we have, this is monomeric protein, and these are the proteins which are all are in monomer, and these are the, we can check. So all these are matching proteins. Okay, and also we can check sequence similarity. So this is the protein with similar proteins, and alignment. So these are the top two proteins, PLXE and SK, which we selected. So we can select and check that where these amino acids are aligned with these proteins. Like here, go to template and select which proteins you want make alignment. Like here I am going to make alignment of only, not this one. So I am going to make alignment of only this protein, which is 100%, and then we go to alignment. So here is the alignment of our protein with the target protein. So in this way we can select our target, and we can check that which proteins are, how many proteins are going to align with this protein. We can also have more options for deep view, and some other options are available. So here is our template, and then we can check our amino acid residues, quaternary structures, sequence similarity with this one. So this is now with this protein, alignment with this protein, and we can go to summary. So this is the summary. And here we have models. Templates. Okay, in this way we get the structure, and Now when we click one of these structure, we can go into that structure. Like here I have clicked this structure for WR 7.1 and we get the detail of this with PDB coordinates and then citation of this and the peptide and these are the amino acid residues. Like here I am going to check that some amino acids, like here I am going to select this isoleucine, so it will be here, and I am going to check this glutamate, it is present here, and then I am going to check that where is cysteine, serine, so it is here. So in this way we can check which amino acids are present, and if there is some amino acids which are not going to match, we can check these amino acids and we can select these amino acids from this sequence, and it will be shown here. Now we move on to another tool which is QMEAN, and by this QMEAN, which is also the tool of Swiss model, we go to this QMEAN tool, and by this we can check the quality of our 3D structure of the protein. And from here now I am going to show you that how we will submit our protein. Like here, this is our project name, which is HCA. We can write here 12, which is our project email ID, and here we will select our coordinate file, and we can select any file. Like here I am going to select this for IWZ, and I am going to check this structure. Now it is submit. After some time it will open this protein and structure of protein, and it will give us the estimation of this structure that we have saved in PDB format. Like here, if we have this structure and I am going to save it in PDB format from here, and then we are going to analyze the 3D structure of this protein. So we will go to QMEAN and submit here that protein. So the job is running here. It is QMEAN version 4.3. So here it is our results, which tells us the quality of this 3D structure of this protein, and we can download this processed PDB file, and here we have matching with the global proteins, which is 0.81 plus minus 0.05, and during pre-processing 20 atoms were removed, so the atoms with zero occupancy, and removed one atom with zero occupancy, removing hydrogen atoms, removed zero hydrogen atoms. Then residue lysine 59 is missing one atom, and 1GO 301 is not the standard amino acid, so it is removed, which is ligand. Zinc 302 is not the standard amino acid, and it is removed, which is also present in zinc is present inside the active site of the enzyme. So in this way we will get the refined structure of our protein, and here is the different amino acid residues, and again we can select these amino acid and analyze that which amino acid is present. It gives us local quality score, and we also have a graph which represents the quality. So predicted local similarity to target, so it represents the similarity of the target with some other proteins. So here is the it is maximum is 1 and the 0.8, and some are above 0.8 and some are below 0.8, and here is the amino acid residues which are present here. So it is the local quality estimation of chain A with some other proteins. So in this way we get the quality assessment of our protein structure. And okay, now we are going to build our own protein. If we are going to build our own protein, we will go to Discovery Studio Visualizer, and here we will go to macromolecules from this tab, or we can go to Tools, Macromolecules, Build and Edit Protein. It will be the same. Now we will open this new molecular window from this one, or we can open from this file, new molecular window. We can also open protein sequence window here. Now we are going to add sequence, and we can grow our chain. We can insert sequence, or we can grow our chain. Like here I am going to select methionine, then tryptophan, threonine, lysine, histidine. The sequence is representing here. We can also insert sequence directly. We can mutate some amino acids. And then we can specify, after this we can specify that alpha helix or beta strand. So this is now create structure from the sequence of amino acids. Now this structure has been created, and we will change the display of this. Now it is alpha helix which we synthesized for this sequence. We can also change here, like here we can change the conformation of this to beta strand. So after this, apply conformation. Now it will be in beta strand sheet. So we applied conformation. After building this one, we will go to again left-handed alpha helix, apply conformation. Now it is alpha helix conformation of our protein. So in this way we can build our protein, and then from this sequence we can again check the homology. So I am going to select, this is very short chain, we can make larger chain, or we can build structure from this sequence also, which is present here. And again I am going to show you that how we can go to Swiss modeler. So we will go to Swiss model, start. Here we will insert our sequence. So there is no uniprot KB sequence for this template, so it will not find similar results. So in this way we can make our proteins, but I am going to show you that if we are going to build here our protein, then we can check the structure and also the similarity of this protein with the target proteins. Like here, if I am going to build this structure, so create structure from sequence of amino acid, we will open this sequence from here, protein sequence window, which is now open here. So when we have a open screen window, so we will write here, like here, we are going to select this methionine. Now create structure from the sequence, and now we are opening this one, and we are going to copy this, and here we are going to paste. So now this protein structure is built by pasting this sequence, and this is our protein. I'm going to show you by rotating it. So now this is our protein. I'm going to zoom out this one. So this is our protein that we have built, and it is because it is alpha helix, because we have selected this alpha helix, and in this way it is alpha helix shape. Okay, so in this way we can create our own alpha helices or beta sheets for protein or sequence of the protein, and then we can go to alignment, and here we can search our homology of this protein, or we can also obtain the structure by this estimation of the structure by this QMEAN Swiss model tool. So thanks for watching my video.